Today we shall discuss about epithelial transport and role of glands. Can you hear me properly? Epithelial transport and role of glands. This is our lecture today. If we talk about the epithelial transport, and before moving to the epithelial transport, first of all, you should know where the epithelial cells they are basically present. So epithelial cell line, they line the hollow organs of the tube, mean all the visual organs. They are lined by these epithelial cells, or all the hollow organs of the tubes. They are lined by the epithelial cells, and they regulate the absorption or secretion of the substances across these surfaces or across these membrane. So when we talk about the epithelial surfaces, one surface of an epithelial cell, it generally faces a hollow or fluid filled chamber, and the plasma membrane on this side will be called as luminal membrane. For example, we have this type of the epithelial cell. So one side is basically facing towards the hollow organ or towards the lumen, and the membrane of this side will be called this epithelial cell. The membrane on this side will be called as luminal membrane. This is lumen side, and this is also this can also be called as apical or mucosal membrane. This membrane or membrane on this side, it can also be called as mucosal membrane or apical membrane of the epithelium. When we talk about the plasma membrane of this epithelial cell, or the membrane of this epithelial cell on the opposite surfaces, which is mostly towards the which is adjacent to the blood vessels, it will be called as basolateral membrane or serosal membrane. So these are the two membrane. One side of this membrane of this epithelial cell, it is facing towards the lumen. This will be called the luminal side or the apical or mucosal membrane, whereas the second membrane of this epithelial cell, it is facing towards the blood vessel, or it is present adjacent to the blood vessels or the blood side. So it will be called as second type of the membrane, that is basolateral membrane or serosal membrane. So here you can see in this diagram that this is the apical side, and this is the basolateral side, or this is luminal side, and this is basolateral side. We can also call this as apical side, and this can also be called as serosal side. Okay. So plasma membrane, which is present on the opposite surfaces, are the basolateral membrane, which is also called as basolateral membrane or the serosal membrane. If we talk about the pathways by which the substances they are cross a layer of the epithelial cell, or how the substances they will cross through a epithelial cell, there are two main pathways. The first one is the by diffusion between the adjacent cells of the epithelium. This is called paracellular pathway. So two types of the pathways are there. One is called simple diffusion method, in which the two adjacent cells, the material will be transported between two adjacent cells with the help of this diffusion method, and this pathway is also called as paracellular pathway. And the second pathway, if we talk about the second pathway, in this pathway, the movement into an epithelial cell, either through the luminar side or the basolateral membrane, it is via the diffusion through the cytosol and exit across the opposite membrane. This is called transcellular pathway. So in the second type, the transcellular pathway, there is again the movement into an epithelial cell, either from the luminar side or from the basolateral membrane, and the molecule, they will diffuse to the cytosol and exit to the opposite side. This is called this will be called as transcellular pathway. So these are two types of the pathways by which the molecules they are moving. In the first example, if we look at the movement of the sodium, here is active transport of the sodium across an epithelial cell. Transepithelial transport of the sodium, it always involves the primary active transport, and in case of primary active transport out of the cell across one of the plasma membrane. So in this case, here you can see that movement of the sodium into the cell across the plasma membrane on the opposite side, it is always downhill. So if we look at the movement of the sodium from this side, the movement is always downhill, mean from higher concentration to from lower concentration. Sodium is always in high concentration on the outer side, and it will move from the outer side to the inner side. This is movement from outer side to the inner side. And on the opposite side, if you look at the movement of the sodium on the opposite side, this moves via the process of primary active transport, where there is direct utilization of the energy in the form of the ATP. So sodium will move from its lower concentration to higher concentration with the help of sodium pump, and that movement will be involving the utilization of the energy. So from the luminal membrane into the epithelial cell, sodium will move via the process of diffusion, and from the epithelial cell to the opposite side or through the basolateral membrane, the sodium will move via the process of active transport, and in this will be primary active transport. So if we talk about the diffusion, simply we can also call it as movement of the sodium via the sodium channel. It can also be called as diffusion. That sodium is basically, in the first case, from the luminal side via the this apical membrane. The sodium is actually moving from its higher concentration to lower concentration, which is also called as diffusion. And from this epithelial cell, or from this intracellular concentration, sodium is again moving from its lower concentration to higher concentration. This is downhill movement from higher concentration to lower concentration, and this is again uphill movement from lower concentration to higher concentration. So it means from the luminal side through the luminal membrane or luminal membrane, through the apical membrane, the sodium will move via the process of diffusion with the help of sodium channel from its higher concentration to its lower concentration, whereas in the intracellular side or inside the epithelial cell, there is always low concentration of the sodium, and sodium will move from its lower concentration to higher concentration through the basolateral membrane with the help of sodium pump, and this movement will be from low concentration to high concentration, and this movement will also involve the utilization of the energy. So this is that's how the movement is actually carried out. This is one example. In some other cases, in some other epithelial cell, the downhill movement sometimes it can be via the secondary active transport. In this case, it is simply diffusion, but in some other cases, this downhill movement it can be carried out with the help of secondary active transport. So the diffusion between the adjacent cell, that is called paracellular pathway, and the movement into an epithelial cell across either the luminal or basolateral membrane, diffusion through the cytosol and exit across the opposite membrane. This is termed as transcellular pathway. So in this example, this is basically when we talk about the movement of the sodium, this is basically transcellular pathway. That sodium is moving from its higher concentration to lower concentration, and again it is moving from its lower concentration to higher concentration. But when we simply talk about this movement, this is called paracellular pathway, mean between the two adjacent cells. This is the lumen side, and this is the epithelial cell. This movement is called paracellular, and this movement, when it moves inside from one end and it moves outside from the opposite end, this will be called as transcellular pathway. So these are the two types of the pathways. If you talk about the diffusion through the paracellular pathway, it can be limited by the presence of tight junction. As all of you know that in certain cells, and specifically in case of plant cells, there are tight junctions between the adjacent cells. So these junctions, they will form a seal around the luminal end of the epithelial cell. But still, some iron, some water, they are able to, they can move, they can diffuse to some degree through this tight junction, and the amount of paracellular diffusion is basically limited by the tightness of the junctional seal and small area, relatively small area available for diffusion. Sometime due to the leakiness nature of this paracellular pathway, and this leakiness nature, it varies in different types of the epithelium. So some may have, some are very leaky and some are having very tight junction. So movement or diffusion depends upon the nature of the tight junction, whether they are leaky or they are tightly arranged. But if we talk about, so it means that during the paracellular pathway, because of the presence of the tight junction, the movement via the process of diffusion or through any other process, that is limited, and this movement actually depends upon the nature of those tight junctions, whether they are leaky or they are tight. But if you talk about the transcellular transport, or in case of transcellular movement, the movement of the molecule through the plasma membrane of the epithelial cell, it will occur via the process of diffusion, and sometimes it can also be carried out via the process of mediated transport, which we have already discussed. And transport and permeability characteristic of the luminal and basolateral membrane, they are not same. If you look at these two membranes, it means the permeability properties of this luminal membrane, it will be different from this basolateral membrane. So these two membranes, they don't have the same permeability, means they will allow the movement of the material to pass through them at different rates, because both of them, they are having the different permeability properties. So these two membranes, why they are having different permeability properties? Because they are different number of the ion channels. Similarly, they are having different number of the transporter proteins. So with respect to the number of the ion channels and with respect to the number of the transporter proteins, these two membranes, they are different from each other as far as their permeability properties are concerned. So as a result of these differences, substances, they will move through these membrane at a different rate, and they can undergo a net movement from a low concentration on one side of epithelium to a higher concentration to the other side. Or in other words, it will move, or it will undergo the active transport across the overall epithelial layer. So if we talk about overall epithelial layer, here you can see that this material is moving from higher concentration to lower concentration through the diffusion, and ultimately this is moving from lower to higher towards the across the basolateral membrane and towards the blood side. So it means, if we talk about overall movement in this case is active transport. Overall movement in this case is active transport, that molecule from the intracellular side, it is moving towards the blood side via the basolateral membrane with the help of active transport. So overall mechanism in this case, that will be considered as active transport. Here, another example, as I have told you in very first slide, that sometime this sodium can also move via the process of secondary active transport, in which the sodium will be behaving as co-transporter. As all of you know, that in case of secondary active transport, sodium can either move with the saluted with the transported solute, or it can move in opposite direction. In one case, it will be called as symport, whereas in other case it will be called as antiport. So another type of the transport of the sodium is the absorption of the material from the gastrointestinal tract into the blood. So the movement between of the substances between the kidney tubules and the blood, as all of you are familiar with this mechanism, that over nephron and over glomerular portion, they are basically performing the, and lobe of Henle, they are performing the reabsorption. So similarly, secretion of the salts and other fluid by the glands. So all these substances, they are reabsorbed by or kidney portion. So in this case, if you look at this diagram here, you can see that active transport across an epithelium, sodium is actively transported across an epithelium from luminal to the blood side. Here you can see that sodium is actively transported from luminal side to the blood side. In this case, it is via the process of secondary active transport, and in this case, it is via the process of primary active transport. So overall, in this diagram, here you can see that sodium is actively transported from luminal side to the basolateral side, or from the luminal side to the blood vessel side via the luminal membrane, and also on the opposite side via the basolateral membrane. So in this case, from blood side to the lumen during the secretion. So movement of the sodium from the lumen into the epithelial cells, it occurs by diffusion through sodium channel in the luminal membrane. We have already discussed that in previous example. The movement of the sodium into the epithelial cell, it can take place via the process of diffusion. So in the second case, the epithelial transport of the organic solute. For example, if we denote that organic solute by X, it will move into cell through a secondary active transport mechanism. And in case of secondary active transport mechanism, as all of you know, that sodium will be moving from its higher concentration to its lower concentration, whereas solute will move from its lower concentration to its higher concentration. So organic substance, it will ultimately move out of the cell at the blood side via the down concentration gradient. Here it is moving from its lower concentration to higher concentration, and when it enters into the epithelial cell, it will move towards the blood side via its down concentration gradient. Mean it will be moving from its higher concentration to lower concentration, from the epithelial cell to the basolateral membrane, with the help of facilitated diffusion or the transport. And it is organic molecule, organic solute, it is large molecule. For the transportation of this large molecule, there will be transporter protein or the carrier protein. So this type of the movement is mediated transport or facilitated diffusion. So it means in this example, sodium will be, yes.
Sir, why it is active transport when it moves from epithelial cell to blood cells?
Because it is from its lower concentration to higher concentration. This solute is present in lower concentration on the epithelial side, and it is present in higher concentration in the epithelial cell. So from the luminal side to the epithelial cell, the movement is of the active transport nature, and this is secondary active transport. This solute is moving along with the company of the sodium, which is moving from its higher concentration to lower concentration. But if you look at the movement of the sodium here, it is moving via the through its down concentration gradient, mean from its higher concentration to lower concentration. But from the epithelial cell towards the blood side, sodium is moving from its lower concentration to its higher concentration. So this is active transport, movement of the sodium, and here this is active transport. So active transport is actually denoted by red arrow, whereas the diffusion process or the mediated transport is denoted by the black arrow. So that's how the organic solute will be moving from its higher concentration to lower concentration through the apical membrane or through the luminal membrane, and it will again be moving. This organic molecule, it is moving from its lower concentration to higher concentration through the luminal membrane, and once it enters into the epithelial cell where it is already having higher concentration, it will start to move from its higher concentration to lower concentration from the basolateral membrane towards the blood vessel. So that's how this process is accomplished across the epithelial cell from the luminal side to the blood vessels towards the blood side. Sodium, if we talk about the sodium will be diffuses into the cell because the intracellular concentration of the sodium is always low. As all of you know that the sodium which enters to the cell via the process of diffusion, that will be simultaneously transported out via the process of active transport with the help of sodium potassium ATPase pump. So once the sodium, it will be entered from the luminal side to the epithelial cell, that sodium will simultaneously, it will be transported from epithelial cell towards the vessels through this basolateral membrane via the process of primary active transport. So overall, the concentration of sodium will be low on inside the epithelial cell, and it will be high on the extracellular side. So sodium moves downhill into the cell, and then uphill out of it. Overall net result, if we talk about the net result is that sodium can be moved from lower to higher concentration across the epithelium. So here this is the net result, that initially the sodium will move down its concentration gradient from high to lower concentration, and now from here it will be moving from its lower concentration to higher concentration. So net result of the sodium movement from the luminal side to the basolateral side is it is active transport, that it is moving from its lower concentration to its higher concentration. Okay, Ji. We have already discussed this figure, that this is the absorption of the organic molecule across an epithelium. Here, organic molecule, which is denoted by X, it is actively transported in the epithelial cell, and epithelial cell is actually absorbing this organic molecule from its lower concentration to its higher concentration. Okay, Ji, if we talk about the organic entry of organic molecule across the luminal plasma membrane, it will be via the active transport, and it is in case of active transport, as all of you know, that secondary active transport mechanism is linked with the movement of ion, and ion in most of the cases is sodium. Sodium, most of the cases, it accompanies with the moving solute particle. So movement of the ion will always be from lower concentration, whereas movement of the solute will be from lower to higher concentration. So in this process, X will move from lower to higher concentration, as X is any type of the ion, any type of the organic molecule, it is moving in the luminal fluid to a higher concentration in the cell, or an exit of the substances across the basolateral membrane. If we talk about the exit of this X on the basolateral side, this X will because it is large, it cannot cross through this membrane via the process of simple diffusion. So because of its large structure, this molecule, this organic molecule, which is denoted by X, it will require certain type of the transporter protein to move through this basolateral membrane. So X will move with the help of carrier protein, which is called mediated transport, or which is called facilitated diffusion, and this X will be moving from its higher concentration to a lower concentration. So overall, if we look at this movement, overall the movement of this X is from its higher concentration to lower concentration, whereas movement of the sodium is from its lower concentration to its higher concentration. So if you look at the concentration of the substances on the blood side, sometimes they can be higher on the blood side and in the lumen, because blood side concentration can approach the equilibrium with the high intracellular concentration with the help of the there can be equilibrium, diffusion equilibrium on the both side, on the two side, but sometimes the concentration of the substances on the blood side, it can be higher. If we talk about the water, as all of you know, that it is not actively transported across the membrane. So net movement of the water and across an epithelium, it can be via the process of osmosis. As all of you know, that we have already discussed the process of osmosis, that with the help of aquaporins, the water molecule, they move from one side of the cell to the other side, and this movement of the water sometimes it can also be accompanied with certain type of the solute particle. So in this case, the movement of the water will be via the process of osmosis, and it can be as a result of the active transport of the solute, especially if you look at the sodium across the epithelium. So here if you look at the movement of the sodium, because all of you know that water actually follows sodium. Where there is higher concentration of the sodium, water will always follow. आपने देखा होगा, आपको जब पसीना निकलता है, आपका जो है वो कम होना शुरू हो जाता है कंसंट्रेशन, तो आपको जो है वो प्यास ज़्यादा फील होती है, या अगर आप कोई नमकीन चीज़ ज़्यादा लेते हैं तो you will have more requirement of the water. Means that water will actually follow the sodium. So when the sodium is moving via the active transport from this interstitial cell to the to the blood side, it means there is always a high concentration of the sodium, and when the sodium concentration in basolateral side will be increased, water will follow the sodium, and water will move via the process of osmosis. I mean, water will move from its higher concentration to lower concentration via the process of osmosis. So water will always follow the sodium. Where there is higher concentration of the sodium, water will follow it. So active transport of the sodium will result in decrease in the sodium concentration on one side of the epithelial layer, on the luminal side, and increase on the other side. Mean when there is active transport, definitely it means that overall the sodium concentration is less on one side and it will be high on the other side of the lumen. So when there is change in the solute concentration, that will be accompanied by the water changes in the water concentration on the two sides, because a change in the solute concentration, it will also induce or produce a change in the water concentration. So here in this case, you can see that this is luminal side and this is, we are having an epithelial cell. As all of you know that in case of sodium is moving from the luminal side to the epithelial side via the process of simple diffusion from its higher concentration to lower concentration. So it means when here the sodium concentration will increase, water will also follow, water will move from its higher concentration to lower concentration. In case of tight junction, the water will move through these tight junction and it will enter into the cell. So when the sodium is actively being transported from epithelial layer towards the blood side, water will again move via the process of osmosis. That water will move from its higher concentration to lower concentration. It will accompany the sodium. So in this case, the water concentration differences produce, excuse me, I have to attend a call. Please stay connected.
Active.
Okay, ji, sorry for interruption. We were discussing about the water concentration, that water will follow the sodium, where the sodium will move, water will move in a company of the sodium. So here in this case, when the sodium is moving from its higher concentration to its lower concentration via the diffusion, water will also move to dilute the sodium, to dilute the solute concentration, and water will move via the process of osmosis. Similarly, when this is moving from its lower concentration to higher concentration, water will again follow, because water is not moving via the process of active transport. Water will always move via the process of osmosis. So water will again follow this sodium, and it will move from this side to the other side. So ultimately, the net movement of the solute across an epithelium, it will be accompanied by the flow of the water in the same direction. And if we talk about the epithelial cell, if they are highly permeable to water, large net movement of the water it can occur, and there will be very small osmolarity differences in osmolarity differences. As all of you know that osmolarity is actually the solute concentration. If this epithelial cell is highly permeable to water, and if there is very small difference of the concentration or small net movement of the solute from this side to the other side, it will result in large movement of the water molecule, because of the highly permeable nature of the this basolateral membrane. If we talk about the glands, they will secrete specific substances into the extracellular fluid, or lumen. This is the next portion, that glands, they are also having their role in the movement of the material. So gland, they will be secreting specific type of the substances into the extracellular fluid, or the lumens of the ducts, in response to the stimuli. When any type of the stimuli is applied to the glands, they will secrete a specific type of the substances. And if you look at the mechanism of the glandular secretion, it will be dependent upon the principle of the membrane diffusion. As all of you know that in case of diffusion, the movement is from higher concentration to lower concentration, or it is of the mediated type, in which the movement is again from higher concentration to lower concentration, but with the help of the transporter protein, or it can be of the exocytosis type we have already discussed. So glands, they are formed during the embryonic development, and glands are formed from the epithelial layers of an organ surface. Mostly glands are, as all of you know, that they are basically formed from the enfolding of the epithelial layer of an organ surface. And glands, they will be, as all of you know, there are two types of the glands. One are endocrine glands, and other are called exocrine glands. So if the glands are connected to the ducts, to the epithelial surface, which they are formed, sometimes they lose their connection, and they may be present in isolated form. So here is, you can see that uh, this is an epithelial cell, and these are the lumen, or these are the duct, and these are the exocrine gland. Similarly, this is endocrine gland. This endocrine gland, it will, as all of you know, that exocrine gland, their secretion into ducts, whereas hormones or other substances secreted by endocrine glands, they diffuses into the blood. So endocrine gland, they will release their secretion directly into the blood, whereas exocrine gland, they will secrete, or they will release into the their secretion into the ducts. So exocrine and their secretion flow through the ducts, and they are discharged into the lumen of an organ. Or in case of skin glands, if we talk about the skin glands, their secretion may be released. For example, sweat glands, their secretion will be released on the surface of the skin. Sweat glands and the salivary glands, they are two examples of exocrine gland. In the second type, if you look at the second type of the glands, they are called endocrine gland. They are ductless gland. Their secretion are released directly into the interstitial fluid surrounding the gland cells. So their secretion will be in the interstitial fluid, and that will be surrounding the uh, their gland cells. So if we look at the movement of the material, material will be diffused into the blood, and once the material is diffused, for example, in case of endocrine gland, their secretion is released into the blood, and from there the blood carries the secretion to all part of the body. Endocrine gland, they release the messengers, those are called hormones, and endocrine gland, they are also called as hormone secreting gland. And it's also important that these are ductless gland. Sometimes they also release non-hormonal substances. For example, if we talk about the liver sometimes secrete organic substances, for example glucose, amino acids, fats and proteins into the blood as well. So in addition to its hormonal function, liver, which is considered as gland, sometimes it also perform non-hormonal function as well, and it is producing organic substances which are non-hormonal type organic substances into the blood. For example, it will secrete glucose, it will secrete the amino acids, fats and proteins as well into the blood. So when these substances are secreted by non-endocrine glands, they will be they will serve as nutrients for other cell to perform specific function in the blood, but they will not be acting as messenger. That's why those will be called as those are not hormones basically in nature. So the substances which are secreted by the glands, they can be into two chemical categories. One are called organic material, that are for the most part they are synthesized by the gland cells, and the second substances which are released by the gland cells, those are salt and water. And those salt and water, they will be moving from one extracellular compartment to the another extracellular compartment, glandular epithelium. So salt and water, they will come from the blood, which is supplying the tissues. Basically, the salt and water will be absorbed by the blood, and ultimately it will be supplied to the tissues. Organic molecules which are secreted by the glands cells, as we have previously discussed, that they will be moved via the various types of the pathways. Diffusion in case of lipid soluble material, if the organic molecule is of non-polar nature, it will simply diffuse across the membrane, and if it is of polar nature, it will be mediated transport, as well as in some cases it will be exocytosis for large molecules. For example, if we have proteins, salts will be actively transported, as we have already discussed in case of sodium, that salt will be actively transported across a glandular epithelial cell, and it will produce the change in the osmolarity as well. So when there is change in the osmolarity, as all of you know, that when there is change in the salt concentration, water concentration will also follow that. So when in case of glandular epithelial cell, when the salts, they are moving from lower concentration to higher concentration, water will always move, and water will follow the salt. In exocrine gland, if we talk about their secretion, their secreted fluid pass through the ducts, and those ducts are basically connecting the glands to the luminal surfaces. So composition of the fluid which is secreted by the gland, that may be altered. This is exocrine gland, and their secretion is directly poured into the lumen of this epithelial cell. So their secretion of the secretion of the exocrine gland, that can be altered because of the absorption or secretion of by the duct cell itself. Sometimes the duct cell, these are the duct cell, they can also change the secretion, they can absorb the secretion produced by these exocrine glands. So this is the difference between the secretion of the exocrine gland and endocrine gland. So composition of the secreted material, if you look at the composition of the secreted material of the exocrine gland, at the end of the duct, it may be it may be different from, and it is also different as far as the rate of the secretion is concerned, and it will reflect the amount of the time the fluid remains in the duct where it can be modified. It means the time the fluid has remained inside in the duct for a larger period of the time, the chances are there that the concentration or the composition of the secreted material secreted by the exocrine gland that may have been altered. So if we look at the rate of secretion, most of the glands, they are having low basal rate of secretion, and in which it means that the secretion can be increased with the help of appropriate signal. Means if there is nerve impulse, hormone or locally generated chemical messenger, it can change the rate of secretion from the gland. But most of the cases, the gland, they are secreted, they secrete their secretion at a very low or the basal rate. The mechanism of increased secretion is, it is again it will also alter the secretory pathway. I mean, when there is increased secretion, the pathway, secretory pathway, that may also be altered. It may involve how the pathway can be altered. It will increase the rate at which a secreted organic substance is synthesized by activating the appropriate enzyme. So when we activate an organic substance is synthesized with the help of an appropriate enzyme, so the rate of the synthesis that can also be increased. Similarly, providing the calcium signal, as all of you know that when there is high concentration of the calcium in the cytosol, it will result in a high rate of the exocytosis. So some the exocytosis of the material can also be increased. The rate can also be increased by increasing the cytosolic calcium concentration. Similarly, pumping rate of the transporter or opening of the ion channel. When the opening and closing of the ion channel or pumping activity of certain sodium potassium ATPase pump that is altered, it will ultimately have an effect on increasing or decreasing the secretion rate from the glands as well. So the volume of the fluid which is secreted by an exocrine gland, it can be increased by increasing the sodium pump activity. As I have told you that when there is increased sodium movement, it will followed by the water movement. So when the water will enter, ultimately the volume of the secretion will be increased. So when sodium and sodium channel, their opening and closing is controlled, as well as the sodium concentration is increased in the exocrine gland. At the moment of the sodium concentration is increased, the volume of the secretion secreted by the exocrine gland can also be increased. And the increased movement of the sodium, it will increase the sodium concentration in the lumen, and when there is increased concentration of the sodium, water will also follow via the process of osmosis. So it means the greater the solute transfer, the greater the water flow as well. Is better. Okay, Ji, this is all about. If you have any question, you can ask, please. Ji, if you have any question, you may ask, please. कोई question नहीं? Okay, Ji, thank you very much. Please stay connected. Okay, जी अनिला Siddiq.
Present, sir.
Your roll number is 3.
Ji, sir.
Then we have Akdas Nadeem.
Yes, sir.
Okay. Atia Rashid. Is she there? Atia. She is
सर उसका नेट
absent.
इशू होगा. नो सर प्रेजेंट है, उसका नेट इशू
Yes,
होगा.
sir. Present.
Okay, Juveria.
Present, sir.
जुवेरिया आपका रोल नंबर क्या है? 011. Is she there, Juveria? She is absent.
No, sir. Present है.
नहीं तो उसका माइक ब्लिंक करना चाहिए ना. अगर वो बोल रही है, वो तो नहीं बोल रही. ये 15, 14 आप लोग बोल रहे हैं.
सर वो क्लास में है, मेरे ख्याल से उसको आवाज नहीं.
Okay. Then we have Maria Savir.
Yes,
Is
sir.
she there?
Present.
Okay.
Yes, sir. Present.
Nazish.
Yes, sir. Present.
Then we have Qazi Sagir.
Yes, sir.
Qazi Muhammad
Present.
Sagir. Okay, your roll number is 17.
Yes, sir. 17.
Okay, then we have Sobia Tariq.
प्रेजेंट सर.
Your roll number is 19.
यस सर.
Okay, then we have Taiba Sijar.
सर सरिश का स्किप कर दिया आपने. स्किप.
Okay, Sarish 18 roll number. Is she there? She is absent, I guess.
ज्वाइन तो की हुई है उसने क्लास शायद अभी.
No, she is not in the list of the participant. And last Taiba Sijar.
Yes, sir. Present.
Okay, ji. Thank you very much. You may leave the session now.